Good afternoon, students. Today we'll be discussing about the medicinal chemistry one for the fourth semester. In the previous classes, I have discussed about the synthesis of catecholamine or adrenergic drugs, and now we'll be discussing about the ACR of sympathomimetic drugs. First of all, we should know what is adrenergic drugs. That means the drug which will stimulate to help in the adrenergic neurotransmitters like adrenaline, noradrenaline. This type of drug will be stimulated and increased their secretion. If there is any problem to secrete the drug, then we will introduce some drug which will mimic the action of the adrenergic neurotransmitter. That means the drug will be having same like action with the adrenaline or noradrenaline hormone. So as they are working on the sympathetic nerves or ANS, that is the autonomic nervous system, that's why they are known as a sympathomimetic drugs. So we have know that the structure contains this type of structure where a benzene ring substituted with the R1 group, whereas one alpha and one beta carbon. So that is the ethyl amine group as it is contained in the ethyl and amine group. That's why this is known as a catecholamine. And the beta position hydroxy group is there. And in the amine group, there is substitution of the R. There may be substitution with the hydrogen or the methyl group, like anything. So depending upon the structure, the changing of the drug is allowing, and the pharmacological activity of the drug will be varying. So the common structural feature required for adrenergic agents are a substituted benzene ring and a primary or secondary aliphatic amino group. That is, the substituted benzene ring will be present, and the primary or secondary aliphatic amino group. Primary, why? Because that may contains the amino group at the primary position, or the secondary carbon that may contains also a hydroxy group. So this primary and secondary carbon will be separated by the two carbon atoms from the benzene. Now in this agent, the class have a hydroxy group in the beta carbon atom, the side chain. So this hydroxy substituted carbon must be in R absolute configuration for maximum direct activity. That means this configuration should be R absolute configuration. As a result, it will having the maximum activity for the adrenergic hormones. Next, substitution in the amino group. That means if there is any substitution in the amino group in this position, the receptor selectivity depends on the size of the alkyl group present in the nitrogen atom. That means whenever the nitrogen is substituted with a very or heavy molecule, then if the alkyl group's molecular size is increased, then the receptor selectivity will be increased. See, increase in the size from the hydrogen in noradrenaline to isoprotronol decreases activity at alpha receptor. That means if there is hydrogen to methyl group or alkyl group is increased, then their receptor activity for the alpha receptor will be decreasing, and the activity to beta receptor will be increasing. So understood? So increase in the alkyl group or the substitution, the alpha receptor activity will be decreased and beta receptor activity will be increased. Now, substitution of the amino group with the tertiary butyl group, like if it is tertiary butyl group, also provide selectivity for different beta receptors. Different beta receptors are present in our different cells or receptors like vasodilators. The beta receptor will be providing the vasodilation. So see, the tarbutaline is selective beta-2 agonist, whereas isoprenaline is a non-selective beta agonist. That means if tertiary butyl group is introduced, then tarbutaline will be acting on the bronchus. As a result, they will enhance the secretion of the sputum, and it also acts in a asthmatic patient also. But isoprenaline, that is one of the important component that can be as a non-selective beta agonist because there is no alkyl group. Now, second carbon is up to the alpha carbon. That means if it is in the alpha carbon substitution, what happens? Substitution on the alpha carbon atom in the side chain. Small alkyl group like the methyl or ethyl may be present on the alpha carbon atom like this. Such substitution slow the metabolism carried out by the monoamine oxidase. That means if there is methyl group or ethyl group present, that means the catabolism of the adrenaline or this catecholamine will be slower down because this substance is degraded by the monoamine oxidase. Now, an ethyl group in this position diminishes the alpha activity and a food compound having beta activity. But if the alkyl group is substituted with the ethyl group C2H5 group, that means all the alpha receptor activity will be diminished and beta receptor activity will be increased. See, metrominol. one of the important drug that is having very good activity, but it is retarded the action of the monoamine oxidase. Ethyl norepinephrine, that is having the beta selectivity very much higher. So substitution on the carbon introduce another asymmetric center producing pair of diastereomers, which can have significantly different biological activity. That means the carbon here becomes a chiral carbon asymmetric center. As a result, it will be producing a diastereomers of this drug. So as a result, the diastereomer R configuration or the S configuration or the D or L, whatever the configuration, they will be having different biological activity. So maximum direct activity in the stereoisomer of the alpha methyl norepinephrine reside with the stereoisomer having 1R2S absolute configuration, where the 1R2R stereoisomer is indirectly showing that activity. That means 1R2S absolute configuration, the two isomer will be 1R2S and 1R2R. So 1R2S absolute configuration having the maximum activity, but the 1R2R having indirect activity. So substitution of the aromatic ring, that means in this aromatic ring, if there is a substitution, then what happens? Compound having both 3,4 hydroxy group in the benzene ring, C, 3, 4, in this position, 3 and 4 are active at both an alpha and beta receptor and they rapidly metabolize COMT. That means if there is substitution at 3 and 4 position, they will having activity at both alpha and beta receptor and they rapidly metabolize by COMT. That is the carboxymethyl transferase. This enzyme will be metabolizing these epinephrine and norepinephrine. As a result, their action will be faster. Change in the substitution pattern from 3 to 5 dihydroxy as in tarbutaline gives good oral activity and selectivity for beta receptor. That means from 4 position to 5 position is the hydroxy group is transferred, then it will be showing good oral activity and selectivity for the beta receptor. So up to this we are ending here. So that means we have to discuss about the amino groups changing, the beta carbons changing and the alpha carbons changing, and the changing in the R configuration for the phenyl ring. Up to this I'm ending here. Thank you all.